新英格兰国际教育源自美国，照耀全球。
So firstly, about the food test. Have you do such experiment at school?
Probably multiple times. I remember, I think, the food test I need to know. There might be some that I don't know still.
Okay.
Yeah.
So we just start with it. It's like in order to test the presence of nutrient, for example, presence of carbohydrates and lipids, we develop this kind of food test by observing the color change. So we say it's called qualitative because we're reading the color change instead of specific data or statistic. And if we're dealing with statistic, that is called quantitative. And do you still remember that we say the qualitative observation is not accurate? So what technique or apparatus can we use in order to make sure the observational color change is more accurate?
Lab tools,
Mm-hmm.
like a colorimeter.
Yep. Very good. So what's the working principle for it?
I don't know how the colorimeter works.
It measures the percentage of light transmission or absorbance, right? That's in one of the work question. And here are the 5 food test solutions that you need to memorize. So the first one is the test for starch. We'll use iodine solution. And since iodine is orange-brown, if there is starch present, the iodine will be changed into blue-black. So we call it positive test result. If the nutrient is present, then the result will be positive. The next one, Benedict's solution, is for sugar, and specifically non-reducing sugar. And then you'll change from blue into brick red. And actually, Benedict's solution has a range of colors, and by observing the specific color, you can tell the concentration for sugar. The next one is ethanol for lipid. It will change from colorless to cloudy, and burette from blue to purple. And there's a new one, it's called test for vitamin C. We'll use DCPIP. Actually, this is the abbreviation for its full name, because the full name is too long to memorize. And the process for it is, you just slowly add drop by drop of the DCPIP into a food solution, and then kindly shake it until it turns colorless. And if it's negative, it's blue. So then we're going to look at the more detailed process. Firstly, you're going to prepare a sample, because most of our food are solid, right? And before we can use this kind of food test, we must change it from solid into solution. So we're going to break up the food using pestle and mortar. It's like to grind the food until all the liquid are squeezed out. And then you transfer them to a test tube and add distilled water in order to dissolve your food and to produce a solution. And finally, you are going to filter the mixture in order to remove all the food that hasn't dissolved or hasn't been grinded. And then after you're getting this pure solution, you can continue with the food test. Does that make sense?
Yeah.
And this one is the Benedict's test. And Benedict's test is a little bit special because it requires warming. So the first step is you add the Bendix solution into your food solution, and then you put it into a water bath to heat it. And after some time, you'll observe color change in your test tube, so you can tell whether the sugar is present or not. And for the water bath, we mainly use a Bunsen burner to heat it up, and to make sure the temperature will be kept around 80 degrees Celsius. So this is the special point for the food test, because for other solutions such as burette or iodine, it doesn't really need heating. Is it clear?
Yeah.
And the next one is for starch iodine. It's really simple. You just prepare a test tube of food sample and then add a few drops of iodine to observe the color change. There's no complicated method.
For all of these, you have to do the initial steps of like stirring and crushing the food, right?
Of course.
Okay.
And the next one is for lipids. And for lipids, we say, firstly, you're going to add the distilled water into the food sample, and then kindly shake it. And then you're going to pour the test tube containing distilled water and food sample into a test tube containing ethanol. And then after shaking, you'll observe the milky white solution. And that's mainly because the ethanol emulsifies your lipids and thus produce the milky white solution.
Yeah.
And for this experiment, there will be a safety hazard. That is, ethanol is highly flammable. So you must make sure that ethanol is kept really far away from naked flames, or you will cause, for example, explosion or a huge fire or something else. Is it clear?
Yeah.
And the final one is about the burette test. It's also really simple, like the iodine test. You just add a few drops of burette solution into your food sample and then kindly shake it.
What does this say A and B like?
Burette A and burette B. Because, well, actually the burette solution composed of two parts, two different kinds of chemical reactants. However, they are not required for you to memorize in GCSE. So you just call it burette solution. And here are some important hazards. And these are called safety hazards as well as precautions, which you might be required to write down in your exams. Firstly, for the burette solution, because it contains copper sulfate, so it's really dangerous. It may cause irritant to your eyes. And therefore, if you're writing the level of the safety hazard, it should be medium or high. And for the precautions, you're going to wear goggles and gloves. And actually, there's a kind of answer that can be applied to almost every question, which is to wear gloves and goggles. And probably some precaution clothes. And then, besides burette, iodine is also irritant, so still wear goggles. And beside these two, which are irritant, there's another one called corrosive, which is sodium hydroxide in the burette solution. So you should wear gloves and also wash your hands immediately if you get some of the chemicals onto your skin. And then it's about ethanol. We say that it's highly flammable, so keep it away from the naked flame, Bunsen burner. And how about if you're dealing with the Benedict's test? Can you think about any safety hazards?
Almost boiling water, maybe.
Yep, very good. So what kind of precautions can you take?
All of them. Could wear gloves when taking the test tube out. Could wear goggles so it doesn't splash on your eyes. I could wear an apron or something so it doesn't splash on your. I don't know what splash anyways, but yeah.
That's very good. So basically, just use like gloves, goggles, or test tube holders. And how about a question like this? I will provide you with a table containing all the results of the food test. And I would like you to write down the conclusion for apple. For example, what kind of nutrients do apple has?
Lots of glucose. Decent amount of iodine. Iodine? Wait, iodine test for starch. There's no fats or and no proteins.
Yep, very good. How about this kind of question?
I mean, it looks like Biarritz for Benedict's for reducing sugars.
Why is it Benedict's?
Because there's a water bath.
Yep, very good. How about this one?
DCPIP turns colorless, right? So no. I think B.
Very good. So what's the original color for Benedict's test?
Blue. So green to brick red. Blue to lilac purple.
Oh, I mean the color change for Benedict test is like from blue to green to orange and finally brick red.
Okay.
New England education. Dream big and make it happen.